So this is just an example of this kind of large-scale biology and what you can do with it. And then I'm going to tell you some of the limitations of it. All right? So what you can do is you can say, all right, I'm going to take, we have the genome of various strains of TB. From that, we have the protein coding regions. We know what the proteins are. We have only 200 out of a total. There are about 4,000 proteins in TB. We have structures. We have three-dimensional configurations for 284, but we can model up to 1,446 because of the fact that many of these sequences are similar to solved structures from somewhere else. So this is called homology modeling, if you're not familiar with it. And so in the end, we can get to about 43% of the human proteome, structurally speaking. Of the TB proteome. We can do roughly the same with human as well. So that's what we know. We know we have this structural complement. And then what we want to do is we want to say, well, what are all of the drug binding sites? If we take all the FDA drugs and we look at everything that they bind to that we know, and we take those binding sites, let's look for all of those binding sites in the TB drugome. All right? So what's interesting, so we're just looking in this database of all protein structures, and it has, many of them have FDA drugs bound to them. In fact, there are 268 FDA drugs in that database, and they're bound to 931 binding sites. And immediately that tells you about drug promiscuity, because if you look at something like methyltrexate, which is known to be a promiscuous drug, it actually binds to 37 distinctly different receptors in this particular database. 37 very different binding sites this drug can bind to. So we're getting this promiscuous effect from a variety of these drugs. So what we're doing then is we're taking all of the proteins in TB and we're taking all of the binding sites and we're making a network of this. And so what we're looking at are the proteins in blue and the drugs are in red. So if I take a point like this, which is a drug, you can see that this is a hub in this network. This is just a visualization of large amounts of biological data, and this is obviously a common technique. And this is a useful case because this particular drug binds to lots of different targets. Why is that useful? Well, it's useful because in principle it means that the chances of an organism becoming, mutating itself and becoming resistant to this particular drug is fairly low. This is all in principle, because there are a whole series of proteins that would have to mutate to actually make it resistant to that particular drug. So what you want to do is you want to take a network like this, then you want to take the same network from, say, human, and overlay them, because the problem is that, this drug might actually bind to all these TB proteins, but it's probably going to bind to a significant number of human proteins as well, which in general terms is probably not a good thing. So what you're looking for is you're looking for instances where you bind as many as possible of the TB receptors and as few as possible as the human receptors. And just by overlaying these networks, you can generate en masse lots of different hypotheses for what would be useful existing FDA drugs that could be used to treat tuberculosis. In principle. All right. So how does it work in practice? All right. So it turns out that one of the high-scoring examples are two drugs called entacapone and tolcapone. So they are actually used in the treatment of Parkinson's disease. They're actually what's called COMT inhibitors. They actually facilitate dopamine derivatives from crossing the blood-brain barrier. All right. So when you go to the literature, you start looking for anecdotal information. Is there any evidence that these two Parkinson's disease drugs have an effect on tuberculosis? And, the problem is, of course, is that hardly anyone is being treated for those two diseases. So, trying to get at that information is difficult. But that's that kind of principle that you would get from actually data mining and text mining the open access literature, if you have access to it, has at least the potential to give you some answers. So anyway, all right, so these are particularly important because they're actually effective against a protein that exists. It's one of these cases when you overlay these networks that exists in TB, but it doesn't exist in human. Therefore, it's a great target. These two drugs appear to be effective against it. So, have we, in fact, come up with potentially an alternative treatment for TB from existing drugs? And of course, repositioning is something you really like and want because you've already been through the FDA process. The drug presumably is considered somewhat safe. And the amount of time and energy and money that goes into having it approved for an alternative use is relatively minimal. So that's the good news. But then this reality comes in, all right? So it turns out that when you look at this, yes, there is binding, there's lots of assay data, but the minimal inhibitory concentration for this drug is actually pretty high. And in fact, so in other words, the amount of drug you'd have to use to get that effect means that it's probably toxic before you get any sign of cure. So that's a problem. And this was work done by the TB Alliance, which is a non-profit organization that's dedicated to the idea of finding new leads for this disease. So you say, oh, that's terrible. Well, yes, it's not what you want, but what it is if you look at the chemical structure of those two drugs, they are different significantly than anything that's been tried against INHA, this particular target, before. So what it represents is a whole new spectrum of potential templates from which to work from. So it's a step, a lot of separate steps. So that's sort of an example. So it all comes from this new way of thinking about polypharmacology, one or multiple drugs binding to multiple targets. This whole notion is also called dirty drugs. And then network pharmacology, which I'll say just a little more about in a second before I finish, is affecting the whole biological network. So I'll give you an example of how you start looking at perturbations to the whole biological network, because we now have inroads into systems biology that allow us to do that. So there was a drug, this is torcetrapib. This was a Pfizer drug. When you go and you see all the empty buildings around here, this in part has to do with it. So torcetrapib was in stage 3 clinical trials. It was going to be the follow-on to Lipitor. It's a cholesterol-controlling drug. And in stage 3 clinical trials, it was discovered that people were dying from mainly caused through hypertension. So it was actually taken out of trials. So literally within a month, the CEO of Pfizer said, this is our follow-on to Lipitor. Stocks started to go like this, came out of clinical trials, plummet. Terrible loss for the company. And essentially it has to do with binding to this cholesterol ester transfer protein. And this is just what it looks like. And it controls the difference between good and bad cholesterol. So the question is, why was the results of hypertension? And we spent quite a lot of time looking at this, because the idea, of course, is that this is actually a pretty good binder to this receptor. So clearly something else is going on to cause this hypertension. Can we identify those targets, modify this drug such that we actually get better binding, we retain the binding to the primary receptor, but whatever the receptors are that are causing the hypertension, we actually reduce that effect. And one of the ways we've been looking at that is the notion of using so-called systems pharmacology